Let's go ahead and talk about the molecular techniques. Here I'm talking about PCR, polymerase chain reaction. So let's get started. Let's first start by having an understanding of nucleic acids. Nucleic acids consist of many nucleotides in a long chain, and these are essential to all known forms of life. There are two types: DNA, which is double helix molecule that carries genetic instructions for growth, development, and reproduction, and RNA, which converts DNA encoded genetic information into proteins. RNA acts as a messenger carrying instructions for DNA for controlling synthesis of proteins. Single-stranded, shorter chains of nucleotides is what RNA is. Okay, humans have 46 chromosomes arranged in 23 pairs. One pair are the sex chromosomes. Okay, the genes are sequences of DNA carried on a chromosome that encode information for the translation of nucleic acid sequences into amino acid sequences that result in the production of proteins. Nucleotides form the basic structure for DNA and RNA. They're linked polymers of pyrimidines or purines, which are base pairs. A polymer is a molecular structure made of similar type bonds. Purines are adenine and guanine, and pyrimidines are cytosine and thymine in DNA, and cytosine and uracil in RNA. Nucleotides are bonded together by hydrogen bonds at the base pair, and two strands are twisted into a helix, and that is in DNA. So a little information on DNA and RNA. In DNA, replication involves one strand of the molecule acting as a template for the creation of the other strand. Hydrogen bonds hold the strands together, forming the helix, but these bonds can be broken, and that's called denaturing or denaturization in the laboratory, such as in PCR. They can also spontaneously be rejoined, and that's called annealing, and functional protein is made from DNA through transcription and translation. RNA is easily replicated, and there are three forms called messenger RNA, which conveys genetic information to the ribosome; transfer RNA, which mediates recognition of the codon, and a codon is a 3 base pair sample and provides corresponding amino acids; and then rRNA, which makes proteins from the ribosome, so that's ribosomal RNA. One of the greatest advances ever in medicine is PCR, or polymerase chain reaction. It can amplify a simple copy of DNA into billions of like copies in 25 to 40 cycles. The principle here is that we have minute amounts of specific DNA which is amplified so it can be further analyzed. A target sequence of DNA must be known in order to use this technology. This sequence needs to be somewhere between 100 and 1,000 base pairs in size. A short portion of that target section of DNA is used as the primers, an even shorter section of DNA, around 16 to 20 base pairs, and these become the template for making of the new DNA copies. And we can use this to analyze products of PCR and gel electrophoresis or real-time PCR using a fluorescent dye. And here is a nice link to the PCR procedure. PCR, or polymerase chain reaction, it occurs in 3 cycles, and this is called the enzymatic process of DNA copying. And the first step in the copying of DNA is called the DNA denaturation cycle. Here, strands of DNA are unwound, and this occurs in about 15 to 30 seconds at 95 degrees centigrade. We have the next stage in the cycle, that is primer annealing, and this is combining primaries with the original single strands of DNA. Here the tubes are placed at 45 to 65 degrees Celsius for about 30 seconds. And then finally we have primer extension. The primers extend DNA with the enzyme DNA polymerase. To make more products, we do primer extension by adding enzyme Taq polymerase at 72 degrees Celsius, and here new nucleotides are added. Having gone through these cycles, specific DNA doubles exponentially. 30 cycles of amplification is typically the mean number of cycles for adequate DNA amplification. There is enough genetic material at this point to analyze through another method like gel electrophoresis, probe hybridization, or direct DNA sequencing, or dot plot and reverse dot plot. There are many methods used to analyze amplified DNA from PCR. We have one method known as the conventional method, which is the use of gel electrophoresis, where bands are separated based on charge. Nucleic acid bands are dyed, then fluoresced with UV irradiation. Then we have DNA sequencing, which is an enzymatic process where complementary DNA to that being sequenced is used to identify the DNA indirectly. We have probe hybridization, which is combining a known labeled nucleic acid probe of genetic material with that derived from the patient or unknown sample. Here we have the formation of hydrogen bonds between nucleotides of RNA and DNA. We have a liquid phase hybridization, where the target nucleic acid and labeled probe interact in a solution. And then we have dot plot and reverse dot plot, used to identify diseases by the analysis of a mutated sequence of DNA. Some other types are Western blot, Southern blot, and Northern blot. These are all methods that we can use to analyze DNA from PCR. Okay? We can use amplification of PCR in areas of the laboratory like microbiology to diagnose bacterial, viral, fungal, or parasitic reactions and infections. And we can also use PCR to determine bacterial resistance. In Western blot, proteins are separated electrophoretically, transferred to a membrane, and then identified through the use of labeled antibodies. In Southern blot, DNA is denatured and the fragments are separated by electrophoresis. Fragments are then added to a membrane and radio-labeled. And then in Northern blot, messenger RNA is separated electrophoretically.